Hello. A huge thank you to the organisers for asking me to give this talk. It's a real pleasure to talk to you today about my career and a little tiny bit of the science that I've been involved in. And mainly what I want to get across is about open-mindedness and being prepared to interact with people from other disciplines to help you to think about your field in new ways, which is something I've done a lot and found really, really interesting. And I hope I can show you today how it's helped me to think about my science in ways that have kept me really interested in it through my career. I started my career as an undergraduate at the University of Warwick. I had been studying chemistry, physics and maths at A-level, and I was best at chemistry, but I thought it was a bit boring. So I went into a programme at Warwick at the time where you could choose from 12 programmes after your first year. And the first year was equal parts biology, chemistry and maths. And then afterwards you could decide which of the 12 available programmes you actually wanted to study in your second and third years. During that first year, I got to know a lot of mathematicians because our walk up and down to our classes was along the same route, and a route that nobody else took. This is because biology and maths at the time shared a building at the top of a hill called Gibbet Hill. To get there, we had to walk past a series of lakes. You can see one of the biggest ones here on the left, and up a path up through woodland, which you can see a picture of on the right, but this is a more recent picture because I'm absolutely sure it wasn't paved in any way when I used to walk up it. On those long walks up and down Gibbet Hill, it's about, not long, 15 minutes, about 15-minute walk, I would spend time chatting to undergrads and postgrads from biology and maths. And I got to know quite a few of the mathematicians. And they introduced me to a lot of ideas which have gone on to influence my career. And this figure shows one of them. This is Waddington's epigenetic landscape, which I like to think of as Waddington's morphogenetic landscape, because I have worked thinking about how organisms develop, how organisms and cells change shape, morphogenesis. This diagram has been used to inspire research in many different fields, and there's a really interesting paper on the history of this diagram and all the different things it's inspired by Bedke in volume 44, issue 4, part B, December 2013, Studies in History and Philosophy of Science, part C, Studies in History and Philosophy of Biological and Biomedical Sciences. This figure really got me thinking about the way that biological processes might work. It shows a marble at the top of a hill, and you imagine the marble is your cell or your organism, and it can roll down the hill. And the point of the figure is that you're much more likely, the cell or the organism or whatever it is you're imagining, is much more likely to take some routes than others. Once it's started rolling down the hill, it's much more likely to go down the valleys than it is up the slopes and over the peaks. And if it wants to go over a peak, it's going to need a lot more energy and a lot more change to get there. The specific example that I'm going to give you today is about root hair development, which is a process I've worked on off and on since the early 90s. Here you can see root hair development in an Arabidopsis root. You can see the surface of the root, and you can see rows of cells that are making these outgrowths, these hairs, and other rows which aren't in between. Every cell in a row of root hairs will make a root hair, but you can see that they're spaced out because the cells are quite long, and they only make their root hairs at one end. They make them at the end nearest the root tip. In the lower image, I've zoomed in using a microscope to show you a whole root hair cell viewed from the side with a little root hair outgrowth towards one end. And this whole elongated cell with a root hair at one end, how this root hair forms just at that one end is the question we're going to address. We're going to think about this from a few different points of view. We're going to think about this root hair cell first as part of a tissue. the skin, the epidermis of the root, which is the cell layer that this little root hair cell finds itself in. We're going to think about it sitting in that organ of the root tip, sitting in the skin of the root, and how that affects where it knows it is and what it thinks it needs to do. But later in the talk, we're going to zoom in and look at the molecules at one end of the cell and think about how the behaviour of those molecules is involved in producing the growth of the root hair. So the first thing that I need to tell you about how this system works is that to make the root hairs, there has to be a signal. A small molecule called auxin needs to be responded to. We can show that an auxin response is needed to make root hairs because we have the experimental tools that we need to turn the auxin response off. So on this slide are images of what happens on the left if we turn the auxin response off permanently so that these cells can never see auxin and respond to it. And on the right, if we turn the auxin response off temporarily using a heat treatment. So here we're using a protein which we're switching on, and with a heat treatment, when it's switched on, it prevents auxin responses. We switch the protein on for 2 hours, and then we allow the protein to switch off again and be degraded, and then we see the auxin response and the root hairs coming back. So on the left you can see that without the auxin response the roots are bald, no root hairs are made, and on the right you can see that as we temporarily turn auxin responses off and then on again, that we see root hairs disappearing and then reappearing on the surface of the root. So what is auxin and how was it discovered? Well, Charles and Francis Darwin saw evidence for the power of auxin when they were looking at things like plants bending towards light signals. And here are some diagrams from their book, The Power of Movement in Plants. Auxin turns out to be a small molecule not that different from the amino acid tryptophan. Here's a diagram of what an IAA auxin molecule looks like, where all of the corners in these molecules are carbon atoms. Auxin moves around the plant through auxin pumps. There are pumps that pump auxin out of cells and pumps that pump auxin into cells. And in between, auxin can diffuse between cell membranes across cell walls. Here you can see some green, where green fluorescent protein has been fused to an auxin out pump that pumps auxin out of cells. It's called a PIN protein. Where the green is, that's where the out pumps are. Lots and lots of copies of that protein, pumping auxin across the membrane. What we find if we look at the tissue carefully, you can see in the image on the left, are lots of little green bands. And in the image in the middle is a close-up which helps us to see that actually the pumps are at the top end of the cells. So here in the image we've got the root tip pointing down and we've got the shoot pointing towards the top of the image. And we're finding these auxin pumps, these out pumps, at the top end of the cell. That means they're pumping auxin out locally there. And there's a picture on the right of Harry Jones, who was at the time my PhD student doing this work, having a look at where auxin pumps were in Arabidopsis roots. To work out what effect this pumping has on auxin in the cells, we needed to do some modelling because there are still not good tools for looking at where auxin actually is in Arabidopsis cells, in fact in any plant cell. There are emerging tools all the time, and there's a lot of excitement about some new tools right now which we're waiting to see how well they work. But certainly at the time of this work, and even now I would argue, there are not straightforward and well-established good, easy ways of seeing where auxin is inside cells. We can see cells responding to auxin at a whole cell level, but we can't see within a cell if one end of the cell has more auxin than another. So to understand what effect the pumps were having, we needed to mathematically model their effect on auxin. And that's what we did on the slide here. In fact, we used a computer model that included mathematical equations. We collaborated with a physicist called Eric Kramer, and we built a three-dimensional model of the Arabidopsis root, and we carefully measured amounts of pump protein that Harry could collect data on by looking carefully in the microscope. We mapped onto the 3D computer model how much pump protein there was based on the fluorescence levels at different places in the 3D map of the root, and then we ran the model to tell us how that predicted auxin moved around the root. To be able to turn levels of pump protein into amounts of auxin moved, we used numbers from the literature where people had measured activities of these proteins in various ways. And we tested how sensitive our model was to changes in those parameters so that we could tell how confident we were about the results. So to cut a long story short, the graph on this slide shows you a summary of what we found out, which is that it's very likely, when we take into account what we know about how fast auxin diffuses, how much auxin these pumps pump, how many pump molecules there probably are based on the fluorescence in the images, we can see that there is a higher concentration of auxin at one end of the cell than the other. There's a higher concentration of auxin at the cell nearest the root tip than at the end of the cell nearest the root tip than at the end of the cell nearest the shoot. You can see on the graph at the top a blue line for the non-hair cells, and at the bottom of the graph a red line for the hair cells, the cells that are going to make root hairs. So because the pumps are pumping auxin out of their top end, their shootward end, that end of the cell has less auxin because it's being shipped out all the time, and it takes time for the molecules to diffuse across the cell, and that's what produces this gradient. To understand the story further, we need to zoom in and think about, instead of the whole tissue and auxin moving through the whole tissue, we need to think about specific molecules inside the cells. So now I'm going to zoom in and look at the local site of growth where the little root hair is forming and think about the molecules that might be in there and what they might be doing. The molecules that we found that locally act on growth are a family of Rho small GTPases called Rho of Plants, or ROP for short. These ROP proteins, we know from lots of different evidence, are coordinating controllers of lots of cell activity in lots of different types of cell. But I'm going to show you some data to try and convince you that they actually can control all the steps of root hair growth. So in the table on this slide, you can see that if we add ROP plus ROP, that means we've roughly doubled the amount of ROP small GTPase protein in the cell. We can change the lengths of root hairs. We can almost double it. And you can see that in the data in the table. But ROP doesn't just change how long the root hairs grow, it also changes how many attempts at making a root hair each cell makes. And you can see that in the image on the bottom. So there you can see microscope images of individual cells, the wild type, which normally only makes one attempt at making a root hair, WT for wild type, which has normal genes, and then the plus ROP, which has double the amount of ROP in each cell. And those plus ROP cells make lots of extra attempts at root hairs, up to 7 we've seen attempts. That means that extra ROP protein is enough to switch on the beginning of the growth process. And the fact that the extra ROP also gives you longer hairs shows that it can also drive more growth as well as starting extra growth. To help with the next stage of the story, I need to show you a video in real time, not speeded up, of what's happening inside a root hair cell. And that's what's on this slide. You can see how old I am from this video because I recorded this a long time ago. This is a loop, so it's the same little bit of video going round and round. As I say, it's not speeded up. So you can see, what I want you to notice is that little bits of the cell are moving around. So the contents of cells are not still. Plants look very stationary from the outside, but they're very much alive on the inside. Lots of stuff is moving around inside them. What does that movie have to do with these Rho small GTPases, these ROPs, which I'm telling you about? Well, we know from lots and lots of different studies of Rho small GTPases in lots of systems that they often cycle between two forms. There's often an inactive form, which is bound to GDP, and an active form bound to GTP. And the active form often has a bit of lipid attached to it. And that's important because that little bit of fat, that little bit of lipid, makes that molecule stickier, which slows it down a bit as it, or actually can be a lot, as it moves around the cell. Now, we made a really interesting observation about ROPs, which is they form in patches where auxin concentrations are high. So I already showed you that we know that the auxin levels are higher at the end of the cell nearest the root tip. And if you look in the bottom left corner, the green image, you can see bright patch of ROP fused to green fluorescent protein, which is why we can see it as green, forming a patch near one end of the cell, near the end of the cell, which is closest to the root tip, at the location where the root hair is about to form. So for somehow there seems to be a relationship possibly between the auxin levels and the formation of the ROP patch. The killer experiment, though, to show that there wasn't a relationship wasn't done by us. It was done by Marcus Graeber's lab. They found that if they disrupted auxin synthesis and transport, they could change where the local high levels of auxin were in roots. So they could make a version of the plant where auxin synthesis and transport didn't work properly. And then they could apply auxin at the wrong end of the cell where the root hair doesn't normally form. And if they did that, they could see the patch of ROP appearing, and then in older cells, the root hair growing at the wrong end of the cell. This was a really, really important result because it really shows that the local concentration of auxin is controlling where the ROP patch forms. And so we started thinking about different ways that the auxin might do this. So let's summarise what we know. We know that there's an auxin gradient across the cell. We know that one end of the cell has higher auxin and the other has lower. And we know that there's a gradient between the two because of auxin diffusion. We knew from work at the time, we knew this from work in other organisms, but now we know it from measurements in plants as well, roughly how fast inactive ROP and active ROP move around. So the GDP bound form and the GTP bound form. And so that meant we could think about and test possible effects of auxin on ROPs by doing some modelling. So we built a simple model of how the ROPs and auxin might interact. We thought about the GTP bound form, which we know moves very fast in cells. We thought about the GTP bound form, which we know moves more slowly. We thought about the arrival rates of those two forms locally at different points in the cell, and the removal rates, and the fact that these two forms can convert between each other. So the GTP bound form, which is in red on this slide, can lose a phosphate and become the GDP bound form. And then there can be a conversion back from GDP bound, which is in blue on the slide, back to GTP bound. We used mathematical approaches and computer modelling approaches to test every possible part of this system where auxin might have an effect that could produce patches of active ROP, like the ones we think we see in experiments. And there are only two aspects of this model where the auxin could influence the model in a way that would produce patches. One is shown in green on this slide. It's the acceleration of the conversion from blue to red. So auxin acting to accelerate the effect that red has on the conversion from blue to red. The second is the arrival of blues locally at this part of the cell. So local arrival of the inactive form. Those are the only two where auxin can have the effect. And the first one, the one that's shown in green on this slide, is more robust and is more likely to be right because it works for a wider range of possible parameters, a wider range of values for auxin concentration, ROP concentration, and so on. So how can we, I've just given you a big statement. I said, we tested where the auxin worked in all these different places, and it definitely only works in these two. But that's not very convincing, is it? You've just got to trust me. So I want to show you how this model actually works by showing you it running. And I can show you what it does in two different ways. So firstly, I'm going to show you an agent-based version of this model. That's where we use a computer, and it's going to use blue dots to represent the ROP GDP, which can move fast. Red dots represent the ROP GDP, which can move slowly. And it's going to allow these to switch colours at low frequencies. And if a blue is near a red, it's going to be more likely to turn red. And the chance of changing blue to red is going to be higher when there's more auxin, and the auxin is going to be shown in green on the model as it's running. And there'll be a little bit of loss of random reds and a little bit of appearance of random blues. Here you can see the model running. You can see the blues and the reds. Reds will appear in a minute, and the green background for the auxin. And you see patches of red appearing as expected. So the agent-based model produces patches of ROP in the expected time frames that we would expect to see at the frequencies and locations we would expect to see based on the experiments. And this was interesting because this model is actually a version of a model proposed by Alan Turing in the early 1950s. You have probably heard of Alan Turing because there was a big celebration of his life in 2012, and there was a famous film, The Imitation Game, starring Benedict Cumberbatch. He did a lot of interesting things in his life, inventing binary code, helping to break the Enigma code. But what I'm talking about today is some work he did in the early 1950s where he thought about how the diffusion of molecules in biological systems might produce the sorts of patterns that we see. He was inspired actually by much larger patterns than the ones I'm talking about today. He was thinking about patterns on things like zebras and fish. But actually it looks as if his systems might have had more relevance to things like the insides of cells, perhaps. That's an idea that is being tested at the moment. Turing's maths is actually not very difficult to follow. And there's a summary on this slide, but if you don't like equations, you don't have to get your head around this to understand my talk. I've put the equations here in case you really like this kind of maths and you happen to have studied it. But if you haven't studied differential equations, or even if you have and you don't really like them, that's fine. If you have, briefly, for those who don't mind equations, you can see that this equation calculates the local amount of active ROP and how that changes with time by considering how fast the form of ROP moves, the conversion rate of one form of ROP into the other, and the loss of ROP from the system plus the arrival of ROP into the system, and does a similar thing for the inactive ROP. And then there's a term which we've applied auxin to, where we consider the way that the active ROP can accelerate the conversion of inactive ROP locally. If you use this equation to look at the model in mathematical terms, rather than as an agent-based model, this can give you a bit more confidence about the results. So I showed you one movie of the agent-based model running, and you might think, oh, yeah, well, that's all well and good, but how do I know you didn't just pick the one time that it gave a patch at the right place? Well, maths allows us to effectively, when we solve a mathematical equation, we're effectively running the model tens of thousands of times and seeing on average what it would do. That's effectively what this kind of maths does for you. So we can plot what this maths predicts would happen. And it shows that if we had a constant level of auxin across the cell, we would find evenly spaced patches of ROP distributed across the cell. But if we have an auxin gradient, which is what we believe we have because of the localized auxin outpumps, then the model predicts we will find patches of ROP near one end, near the end where the auxin is high. And that's where we should find root hairs too. So the model predicts what we actually see. Now we can go a stage further and plot a graph. I wish this was as pretty as Waddington's epigenetic landscape, but it really isn't. However, I hope you can see what it shows. What we've plotted here is the length of the root hair cell versus the auxin concentration. And it tells you, for a given cell with those properties, what phenotype we would expect to see. So it's a map. This graph is a map, and you could measure cell length and you could measure auxin concentration, and it would tell you what you'd expect what the phenotype to be in terms of numbers of root hairs and where you would find them. So the trajectories on this show you how a cell might pass through this as it gets longer and as its auxin concentration changes. And we can see different regions on this graph where we might find a boundary patch, that's a root hair at one end, an interior patch, that's a root hair a bit further into the cell, one of each, and so on. I wanted to mention the people who did this work with me. So this is Robert Payne, who helped me to develop the simple ROP model I've been talking to you about. And one of the nice things that came out of this modelling is that it makes really strong predictions that we could go on and test. And one of those predictions was that the maths model, and Victor Brenier showed this as part of his PhD, which won a prize actually in the Faculty of Engineering at the University of Bristol. Victor showed that the maths predicts that the patch actually forms right up against the end of the cell and then should drift along. It should drift with time away from the end of the cell. So this was predicted by the maths. When we went to have a look in experiments, Harry had a look in her experiments, and she found, so Harry's on the right, Victor's in the middle. Victor's other supervisor, Alan Chapney's, is on the left. Harry found in the experiments, looking very carefully in the microscope, that the ROP patch does in fact move exactly as Victor's maths predicted. So the story's not sewn up yet, but I think we've got a long way. Why does a root hair form at one end of the cell and not the other? And the answer is because ROPs drive local growth, and an auxin gradient controls where a ROP patch forms. And by working with mathematicians and engineers and computer modelers and physicists, we've been able to do modelling which suggests that the auxin probably functions by locally altering the ROP activity and mobility in the cell, and that in this way a gradient of auxin across the cell can produce a patch of ROP which can then drive the growth of the hair. Thank you for listening.